Chapter 18 Body Fluids and Circulation. You have learned that all living cells have to be provided with nutrients, oxygen, and other essential substances. Also, the waste or harmful substance produced have to be removed continuously for a healthy functioning of tissues. It is therefore essential to have efficient mechanism for the movement of these substances to the cell from the cells. Different groups of animals have evolved different methods for this transport. Simple organisms like sponges, coelacanths, circulate water from their surrounding through their body cavities to facilitate the cells to exchange these substances. More complex organisms use special fluids within their bodies to transport such material. Blood is the most commonly used body fluid by most of the higher organisms, including humans, for this purpose. Another body fluid, lymph, will also help in the transport of certain substances. In this chapter, you will learn about the composition and properties of blood and lymph tissue fluid, and the mechanism of circulation of blood is also explained here. In 18.1, blood is a special connective tissue consisting of a fluid matrix, plasma, and formed elements. 18.1.1 Plasma is a straw colored viscous fluid constituting nearly 55% of the blood. 90 to 92% of the plasma is water, and protein contributes 6 to 8% of it. Fibrinogen, globulins, and albumins are the major protein. Fibrinogen are needed for clotting or coagulation of blood. Globule primarily are involved in defense mechanism of the body, and the albumin help in osmotic balance. Plasma also contains small amount of minerals like sodium, calcium, magnesium, HCO3, Cl, etc. Glucose, amino acid, lipids, etc. are also present in the plasma, as they are always in transit in the body. Factors for coagulation or clotting of blood are also present in the plasma in an inactive form. Plasma without the clotting factor is called serum. 18.1.2 Formed elements. Erythrocyte, leukocyte, and platelets are collectively called formed elements, figure 18.1, and they constitute nearly 45% of the blood. Erythrocyte or red blood cell are the most abundant of all the cells in body. A healthy adult man has on an average 5 million to 5.5 million of RBCs of blood. Red blood cells are formed in the red bone marrow in the blood. Adults RBCs are devoid of nucleus in most of the mammals and are biconcave in shape. They have a red colored iron containing complex protein called hemoglobin, hence the color and name of these cells. A healthy individual has 12 to 16 grams of hemoglobin in every 100 ml of blood. These molecules play a significant role in transport of respiratory gases. RBCs have an average lifespan of 120 days, after which they are destroyed in the spleen graveyard of RBC. Leukocytes are also known as white blood cells, as they are colorless due to the lack of hemoglobin. They are nucleated and are relatively lesser in number, which average 6,000 to 8,000 of blood. Granulocytes and agranulocyte, neutrophils, eosinophils, and basophils are different types of granulocytes, while lymphocytes and monocytes are the agranulocytes. Neutrophils are the most abundant cells. 60 to 65% of the total white blood cells are basophil, are the least, 0.5 to 1% among them. Neutrophils are monocytes. 6 to 8% are phagocytic cells which destroy foreign organisms entering the body. Basophils secrete histamine, serotonin, heparin, etc. and are involved in the inflammatory reaction. Eosinophils 2 to 3% resist infection and are associated with allergic reaction. Lymphocytes 20 to 25% are of two major types, B and T forms. Both B and T lymphocytes are responsible for immune response of the body. Platelets are also called thrombocytes and are cells fragment produced from megakaryocytes, special cell in the bone marrow. Blood normally contains 1,50,000 to 3,50,000 platelets. Platelets can release a variety of substances, most of which are involved in a coagulation or clotting of blood. A reduction in their number can lead to clotting disorder, which will lead to excessive loss of blood from their body. 18.1.3 Blood groups. As you know, blood of human beings differ in certain aspects, though it appears to be similar. Various types of grouping of blood has been done. Two such grouping like ABO and RH are widely used all over the world. 18.1.3.1 ABO grouping. ABO grouping is based on the presence or absence of two surface antigens chemicals that can induce immune response on the RBCs, namely A and B. Similarly, the plasma of different individuals contain two natural antibodies, proteins produced in response to antigen. The distribution of antigens and antibodies in the four groups of blood A, B, AB, and O are given in table 18.1. You probably know that during blood transfusion any blood cannot be used. Blood of a donor has to be carefully matched with the blood of a recipient before any blood transfusion to avoid several problems of clumping, destruction of RBC. The donor compatibility is also shown in table 18.1. Table 18.1, blood groups and donor compatibility. From the above mentioned table, it is evident that group O blood group can be donated to person with any other blood group, and hence O group individuals are called universal donor. Person with AB group can accept blood from person with AB as well as the other groups of blood. Therefore, such person are called universal recipients. 18.1.3.2 RH grouping. Another antigen, the RH antigen, similarly to one present in rhesus monkeys. Hence, RH is also observed on the surface of RBCs of majority, nearly 80% of humans. Such individuals are called RH positive, and those in whom this antigen is absent are called RH negative. An RH negative person, if exposed to RH positive, will form specific antibody against the RH antigen. Therefore, RH grouping should also be matched before transfusion. A special case of RH compatibility mismatching has been observed between the RH negative blood group of a pregnant mother with the RH positive blood of the fetus. RH antigen of the fetus do not get exposed to RH negative blood of the mother in the first pregnancy, as the two blood groups are well separated by the placenta. However, during this delivery of the first child, there is a possibility of exposure of the maternal blood to small amount of the RH positive blood from the fetus. In such cases, the mother starts preparing antibodies against RH antigen in her blood. In case of subsequent pregnancy, the RH antibodies from the mother RH negative can leak into the blood of the fetus RH positive and destroy the fetal RBCs. This could be fatal to the fetus or could cause severe anemia and jaundice to the baby. This condition is called erythroblastocyst fetalis. This can be avoided by administering anti-RH antibodies to the mother immediately after the delivery of the first child. 18.1.4 Coagulation of blood. You know that when you cut your finger or hurt yourself, your wound does not continue to bleed for a long time. Usually the blood stops flowing after some time. Do you know why blood exhibits coagulation or clotting in response to an injury or trauma? There is a mechanism to prevent excessive loss of blood from the body. You would have observed a dark reddish brown scum formed at the site of a cut or an injury over a period of time. This is a clot or coagulum formed mainly of a network of threads called fibrin, in which dead and damaged formed elements of blood are trapped. Fibrins are formed by the conversion of inactive fibrinogen in the plasma by the enzyme thrombin. Thrombin in turn are formed from another inactive substance present in the plasma called prothrombin. An enzyme complex thrombokinase is required for the above reaction. This complex formed by a series of linked enzymic reaction cascade process involving a number of factors present in the plasma. In an inactive state, an injury or a trauma stimulate the platelets in the blood to release certain factors which activate the mechanism of coagulation. Certain factors released by tissues at the site of injury also can initiate coagulation. Calcium ion play a very important role in the clotting. 18.2 Lymph tissue fluid. As the blood passes through the capillaries and tissues, some water along with many small water soluble substances move out into the space between the cells of tissues, leaving the larger protein and most of the formed element in the blood vessel. The fluid released out is called interstitial fluid or tissue fluid. It has the same mineral distribution as that in plasma. Exchange of nutrient gases etc. between the blood and the cells always occur through this fluid. An elaborate network of vessels called the lymphatic system collects this fluid and drains it back to the major veins. The fluid present in the lymphatic system is called lymph. Lymph is a colorless fluid containing the specialized lymphocytes which are responsible for the immune response of the body. Lymph is also an important carrier for nutrients, hormones, etc. Fats are absorbed through lymph in the lacteals present in the intestinal villi. 18.3 Circulatory pathway. The circulatory patterns are of two types: open or closed. Open circulatory system is present in arthropods and mollusks, in which blood pumped by the heart passes through large vessels into open space or body cavity called the sinus. Annelids and chordates have a closed circulatory system in which the blood pumped by the heart is always circulated through a closed network of blood vessels. This pattern is considered to be more advantageous as the flow of fluid can be more precisely regulated. All vertebrate possesses a muscular chambered heart. Fishes have a two-chambered heart with an atrium and ventricle. Amphibians and the reptiles except crocodile have a three-chambered heart and two atria and a single ventricle, whereas crocodile, bird and mammal possesses a four-chambered heart with two atria and two ventricles. In fishes, the heart pumps out deoxygenated blood, which is oxygenated by the gills and supplied to the blood body parts, from where deoxygenated blood is returned to the heart. Single circulation. In amphibians and reptiles, the left atrium receives oxygenated blood from the gills, lungs, skin, and the right atrium gets the deoxygenated blood from other body part. However, they get mixed up in a single ventricle which pumps out mixed blood in incomplete double circulation. In birds and mammals, oxygenated and deoxygenated blood received by the left and right atria respectively passes on the ventricles of the same side. The ventricle pump it out without any mixing up. Two separate circulatory pathways are present in these organisms, hence these animals have double circulation. Let us study the human circulatory system. 18.3.1 Human circulatory system. Human circulatory system are called the blood vascular system. Consists of a muscular chambered heart, a network of close branching blood vessel and blood, the fluid which is circulated. Heart, the mesodermally derived organ, is situated in the thoracic cavity in between the two lungs, slightly tilted to the left. It has the size of a lynched fist. It is protected by a double-walled membranous back pericardium enclosing the pericardial fluid. Our heart has four chambers, two relatively small upper chambers called atria and two larger lower chambers called ventricles. A thin muscular wall called the interatrial septum separates the right and the left atria, whereas a thick-walled and interventricular septum separates the left and the right ventricle. Figure 18.2. The atrium and the ventricle of the same side are also separated by a thick fibrous tissue called atrioventricular septum. However, each of these septa are provided with an opening through which the two chambers of the same side are connected. The opening between the right atrium and the right ventricle is guarded by a valve formed of three muscular flaps or tricuspid valve, whereas the bicuspid or mitral valve guard the opening between the left atrium and the left ventricle. The opening of the right and the left ventricle into the Figure 18.2 section of human heart. Pulmonary artery and the aorta respectively are provided with the semilunar valve. The valve in the heart allows the flow of blood only in one direction, from the atria to the ventricle and from the ventricle to the pulmonary artery or aorta. These valves prevent any backward flow. The entire heart is made up of cardiac muscle. The walls of ventricles are much thicker than that of atria. Specialized cardiac musculature called the nodal tissue is also distributed in the heart. Figure 18.2. A patch of the tissue is present in the right upper corner of the right atrium called the sinoatrial node (SEN). Another mass of the tissue is seen in the lower left corner of the right atrium close to the atrioventricular septum called the atrioventricular node. A bundle of nodal fibers, ventricular bundle, continue from the avian which passes through the atrioventricular septa to emerge on the top of the interventricular septum and immediately divide into a right and left bundle. These branches give rise to minute fiber throughout the ventricle musculature of the respective side and are called Purkinje fiber. These fibers, along with right and left bundles, are known as bundle of His. The nodal musculature has the ability to generate action potential without any external stimuli. It is auto-excitable. However, the number of action potential that could be generated in a minute vary at different parts of the nodal system. This SAN can generate the maximum number of action potential 70 to 75 minute and is responsible for initiating and maintaining the rhythmic contractile activity of the heart. Therefore, it is called a pacemaker. Our heart normally beats 70 to 75 times in a minute, every 72 beats per minute. 18.3.2 cardiac cycle. How does the heart function? Let us take a look. To begin with, all the four chambers heart are in relaxed state. They are in joint diastole and the tricuspid and bicuspid walls are open. Blood from the pulmonary veins and vena cava flows into the left and the right ventricle respectively. Through the left and right atria, the semilunar valves are closed at this stage. The SAN now generate an action potential which stimulates both the atria to undergo a simultaneously contraction the atrial systole. This increases the flow of blood into the ventricles by about 30%. The action potential is conducted to the ventricular side by the avian and AV bundle, from where the bundle of His transmit it through the entire ventricular musculature. This causes the ventricular muscle to contract. Ventricular systole. The atria undergo relaxation diastole coinciding with the ventricular systole. Ventricular systole increase the ventricular pressure, causing the closure of tricuspid and bicuspid wall due to attempted backward of blood into the atria. As the ventricular pressure increases further, the semilunar valves guarding the pulmonary artery right side and the aorta left side are forced open, allowing the blood in the ventricles to flow through these vessels into the circulatory pathway. The ventricles now relax. Ventricular diastole and the ventricular pressure falls, causing the closure of semilunar valve, which prevent the backflow of blood into the ventricle. As the ventricular pressure declines further, the tricuspid and bicuspid valve are pushed open by the pressure in the atria exerted by the blood which was being emptied into them by the veins. The blood now once again moves freely to the ventricle. The ventricles and atria are now again in the relaxed joint diastole state as earlier. Soon the SAN generate a new action potential, and the event described above are repeated in that sequence. The process continue. The sequential event in the heart, which is cyclically repeated, is called the cardiac cycle, and it consists of systole and diastole of both the atria and ventricle. As mentioned earlier, the heart beats 72% time per minute. That many cardiac cycles are performed per minute. From this, it could be deduced that the duration of the cardiac cycle is 0.8 seconds. During a cardiac cycle, each ventricle pumps out approximately 70 ml of blood, which is called the stroke volume. The stroke volume multiplied by the heart rate number of beats per minute give the cardiac output. Therefore, the ventricle per minute and average 5000 ml or 5 liter. In a healthy individual, the body has the ability to alter the stroke volume as well as the heart rate and thereby the cardiac output. For example, the cardiac output of an athlete will be much higher than that of an ordinary man. During each cardiac cycle, two prominent sounds are produced which can be easily heard through a stethoscope. The first heart sound lub is associated with the closure of the tricuspid and bicuspid valve, whereas the second heart sound dub is associated with the closure of the semilunar valves. These sounds are of clinical diagnostic significance. 18.3.3 Electrocardiograph (ECG). You are probably familiar with this scene from a typical hospital television show. A patient is hooked up to a monitoring machine that shows voltage traces on a screen that makes the sound pip 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 pee as the patient goes into cardiac arrest. This type of machine, electrocardiograph, is used to obtain an electrocardiogram (ECG). ECG is a graphical representation of the electrical activity of the heart during a cardiac cycle. To obtain a standard ECG as shown in figure 18.3, a patient connects to the machine with 3 electrical leads, one to the each wrist and to the left ankle, that continuously monitor the heart activity. For a detailed evaluation of the heart function, multiple leads to are attached to the chest region. Here we will talk only about a standard ECG. Each peak in the ECG identified with a letter from P to T that correspond to a specific electrical activity of the heart. The P wave represents the electrical excitation or depolarization of the atria, which leads to the contraction of both the atria. The QRS complex represents the polarization of the ventricle, which initiate the ventricular contraction. The contraction starts shortly after Q and marks the beginning of the systole. The T wave represents the return of the ventricle from excited to normal state repolarization. The end of the T wave marks the end of the systole. Obviously, by counting the number of QRS complex that occur in a given time period, one can determine the heartbeat rate of an individual. Since the ECG obtained from different individuals have roughly the same shape for a given lead configuration, and deviation from this shape indicate a possible abnormality of disease. Hence it is of a great clinical significance. 18.4 double circulation. As mentioned earlier, the blood pumped by the right ventricle enter the pulmonary artery, whereas the left ventricle pumps blood into the aorta. The deoxygenated blood pumped into the pulmonary artery is passed on to the lungs, from where the oxygenated blood is carried by the pulmonary veins into the left atrium. The pathway constitutes the pulmonary circulation. This oxygenated blood entering the aorta is carried by the network of arteries, arterioles, and capillaries to the tissues, from where the deoxygenated blood is collected by a system of venules, veins, and vena cava, and emptied into the right atrium. This is the systematic circulation. Figure 18.4. The systematic circulation provides nutrients, oxygen, and other essential substances to the tissue, and takes carbon dioxide and other harmful substances away from elimination. A unique vascular connection exists between the digestive tract and liver, called the hepatic portal system. The hepatic portal veins carries blood from intestine to the liver before it is delivered to the systematic circulation. A special coronary system of blood vessel is present in our body exclusively for the circulation of blood and from the cardiac musculature. 18.5 Regulation of cardiac activity. Normal activities of the heart are regulated intrinsically, auto-regulated by a specialized muscle nodal tissue. Hence the heart is called myogenic. A specialized neural center in the medulla oblongata can moderate the cardiac function through autonomic nervous system (ANS) neural system. Through the sympathetic nerve part of ANS can increase the rate of the heartbeat, the strength of ventricular contraction, and thereby the cardiac output. On the other hand, parasympathetic neural signals, another component of ANS, decrease the rate of the heartbeat, the speed of conduction of action potential, and thereby the cardiac output. Adrenal medullary hormones can also increase the cardiac output. 18.6 Disorder of circulatory system. High blood pressure, hypertension. Hypertension is the term for blood pressure that is higher than that of normal, 122 by 80. In this measurement, 120 mmHg, millimeters of mercury pressure, is the systolic or pumping pressure, and 80 mmHg is the diastolic or resting pressure. If repeated check of blood pressure of an individual is 140 by 19, 140 over 19 or higher, it shows hypertension. High blood pressure leads to heart disease and also affect vital organs like brain and kidney. Coronary artery disease (CAD). Coronary artery disease is often referred to as the atherosclerosis. Affect the vessel that supply blood to the heart muscle. It is caused by a deposit of calcium, fat, cholesterol in fibrous tissue, which makes the lumen of arteries narrower. Angina. It is also called angina pectoris. A symptom of acute chest pain appears when no oxygen is reaching the heart muscle. Angina can occur in men and women of any age, but it is more common among the middle-aged and elderly. It occurs due to conditions that affect the blood flow. Heart failure. Heart failure means the state of heart when it is not pumping blood efficiently enough to meet the needs of the body. It is sometimes called congestive heart failure because congestion of the lung is one of the main symptoms of this disease. Heart failure is not the same as cardiac arrest. When the heart stops beating, or the heart attack, when the heart muscle is suddenly damaged by an inadequate blood supply. Summary: vertebrates circulate blood, a fluid connecting tissue in their body to transport essential substance to the cells and to carry waste substances from there. Another fluid, lymph tissue fluid, is also used for the transport of certain substances. Blood comprises of a fluid matrix, plasma, and formed elements: RBCs, erythrocytes, white blood cell, leukocytes, and platelets. Thrombocytes constitute the formed elements. Blood of human are grouped into A, B, AB, and O system based on the presence or absence of two surfaces antigen A, B on the RBCs. Another blood grouping are also done based on the presence or absence of another antigen called rhesus factor Rh on the surface of RBCs. This fluid called lymph is almost similar to blood except for the protein content and formed element. All vertebrate and a few invertebrates have a closed circulatory system. Our circulatory system consists of a muscular pumping organ, heart, and a network of vessels and a fluid. Blood. Heart has a two atria and two ventricles. Cardiac musculature is auto-excitable. Sinoatrial node generate the maximum number of action potential per minute, 70 to 75 per minute, and therefore it sets the pace of the activities of the heart. Hence it is called the pacemaker. The action potential cause the atria and then the ventricles to undergo contraction, systole, followed by their relaxation, diastole. The systole forces the blood to move from the atria to the ventricle and to pulmonary artery and aorta. The cardiac cycle is formed by sequential event in the heart, which is cyclically repeated and is called the cardiac cycle. A healthy person shows 72 such cycles per minute. About 70 ml of the blood is pumped out by each ventricle during a cardiac cycle and is called the stroke or beat volume. Volume of blood pumped out by each ventricle of heart per minute is called the cardiac output, and it is equal to the product of the stroke volume and heart rate, approx 5 liters. The electrical activity of the heart can be recorded from the body surface by using electrocardiograph, and the recording is called electrocardiogram, ECG, which is clinically important. We have a complete double circulation. Two circulatory pathways, namely pulmonary and systemic, are present. The pulmonary circulatory start by the pumping of deoxygenated blood by the right ventricle, which is carried to the lung where it is oxygenated and returned to the left atrium. The systematic circulation start with the pumping of oxygenated blood by the left ventricle to the aorta, which is carried to all the body tissues, and the deoxygenated blood from there is collected by the veins and returned to the right atrium through the heart. Is auto-excitable. Its function can be moderated by neural and hormonal mechanism.